feel like I've waited long enough. Maybe too long. How's everybody doing out there? I'm on the Scanning Electron Microscope. You can hear it buzzing in the background. And we're just rolling with some samples from Idaho today, from a power plant. Kind of a just chilling between lessons with your latte. That's nice. I wish we had some sort of I mean, we have like you could go to Starbucks on campus, but like, I guess I just don't want to spend $6 on a tea, you know what I mean? So, I never go there, or almost never go there. I mean, their tea is just Tazo tea. You could buy it at the grocery store, and you could just get some milk and cook it in the microwave and stick some Tazo tea in, and you could, I mean, for $6, you could buy a gallon of milk and a carton of Tazo chai tea. And, I mean, for the price of one, you could make, I don't know, at least 8. So, an AeroPress. Oh, yeah, that's nice. I keep threatening to buy one of these things that you like, a milk frother, but mostly out of just like being funny, because I can't imagine spending $25 to froth milk, because you could just put it in the microwave. But because I mostly just drink chai tea. So this is an image from my SEM, and back when I first started streaming, I did this a few times, and I thought it was kind of fun to start like super zoomed in on something and then kind of gradually work our way out. So the field of view that we're looking at for the current, this is a piece of a diatom that we're looking at. We're currently at a magnification of almost 100,000 times. I think I could actually get a little bit closer if we wanted to actually get 100,000. Let's see, no, that's 125,000 right there. So our magnification's 125,000 times. And, you know, that's relatively easy to do once we're kind of zoomed in already. And then just kind of gradually sort of back away from what we're looking at so that you get a good sense of how ridiculously close we are to this. So the scale bar at the bottom of the image says 500 nanometers. That's what the NM stands for. So, you know, that's like 1,000th of a millimeter is a nanometer, right? And then we have 500 of those. So really, oh no, sorry, that's 1,000th of a micron. So we're at basically that scale is half of a micron, right? Half of a micron. So, and so our total field of view here across the specimen that we're looking at right now is about 3 microns, right? So just moving out to get a sense of what we're looking at. And I see all these crescent-shaped structures that are here, sort of C-shaped structures that are here. And those are actually the pores on the valve face of a diatom. And these little blebs that are here are bits of, I don't know, I suspect they're probably mud, or in some cases they might be silica deposits that the diatoms put on a little bit of extra silica on the valves, but I think it's probably just a little bit of junk. It could also be a little bit of the nitric acid that we use to digest the diatoms, and a little bit of it may precipitate on the actual valve. So just zooming out, so let's see, this is around 35,000 times magnification, so just that little bit of distance is actually really kind of a lot. And now our field of view is about 7.5 microns. So we just kind of like doubled our distance across the field of view. And you can see that there's a whole bunch of these little crescent-shaped structures. They're not all the same. So some of them are kind of C-shaped, and some of them are sort of double C-shaped. And then this one here looks like a Wu-Tang W. And this one over here looks like two anchors stuck in each other, right, from the top. But we can just kind of slowly sort of coast outward. And what you can see is that there's actually a sort of a fabric that shows up when you look very carefully at them. So it becomes more apparent right here that the little areolae that we were looking at, the sort of, here's our, W shape, and here's our two anchors, and these sort of C-shaped or double C shapes, but that they actually line up in rows. And on a diatom, these rows are actually called striae. The striae just is Latin, means stripe. So in the light microscope, they look like stripes, and we never see this actual structure. We might see these as little dots, but we don't ever actually see the C shapes. Like I wouldn't be able to tell in a light microscope that these things are C-shaped at all. I'd have no idea. They just look like little holes in the light microscope, and that's because for the light microscope, it's like an X-ray. It sees through the diatom rather than seeing the surface of it. And when the light passes through these openings, they show up as little bright spots, right? So that's all we would see, so sort of coarse resolution. So if I zoom out a bit farther to 7,000 times magnification, you kind of start to see the actual organism rather than just looking at essentially little pores on the surface of the organism. And we can see this sort of long hole in the valve face, which is the sort of slit-shaped hole. Hello, Mallory.
Hello.
Which is the raphe of a diatom, and we're looking on the outside of it, so an external view. So I just sort of keep coasting outward. Now our field of view is about 80 microns across, right? So we're at the point where we could almost see the whole width of this diatom. We can't see the whole length of it, but you get sort of a sense that it's a fairly large diatom that we're looking at. And all those little holes, this is the way they would basically look on a light microscope, or kind of close to it. They would just be like a series of little holes now. Like you wouldn't actually be able to see much of the context for them. And if we keep just sort of drifting out, again, we can kind of get a better sense of the structure of the organism that we're looking at. So here's the raphe, one branch of the raphe going this way. This is the proximal end for one of the valve raphe branches. And here's the other start of this one, which goes this direction. And we can actually see that down here on one end, the raphe kind of diverts, and it deflects towards the dorsal side, the sort of hump side, like the fin of a whale or whatever would be on this end if it were a whale. Also, we can see there's some other diatoms in our field of view. So we just keep drifting outward. What we'll find is that what we're looking at is sort of a diatom that has a symbol shape, right? So like its total shape is kind of like a loaf of bread on its side, or a slice of bread maybe if you cut it, and or a symbol, right? Like with a hump on one side and a more or less straight or maybe slightly tumescent side on the other side. And this is actually a diatom from the genus Cymbella, so named sort of for the symbol shape that it has. And actually I wanted to come down here and look at this end of the valve where the raphe deflects, because there's a lot of detail that we can see. So now we're zoomed back in. We're looking at around 10,000 times magnification, and you can see the raphe coming down here, and then as I mentioned, it deflects. So we see it basically deflected in one direction, and towards the dorsal side. And that actually is like the key observation that you need to make if you're looking at something that is symbol shaped, to figure out whether it belongs in the genus Cymbella or the genus Encynema. So for Encynema, it would come down and deflect this way, right? So rather than going towards the dorsal side, it would go towards the ventral side. And you can see that some of the pores down here are kind of like an interesting S shape or Z shape. I'm going to tweak the brightness and contrast a bit because it's really bright down here along the bottom. And that'll make it a little bit easier to see some of the detail in here. And then you can also see some of these pores, like in here, are kind of like, they look like letters, right? They're really intricate shapes. So this is what we would call an apical pore field. And for the genus Cymbella, this is required. It must have an apical pore field on both ends, or it isn't the genus Cymbella. It would be something else. And if it had these features and didn't have this, we'd have to give it a different name, which would probably be Delicatula. So we can zoom in again, sort of this area to sort of get a look at these cool little openings. They look very much like little letters. If you didn't know that we were looking at the outside of a diatom valve, you might think, oh, this is cool. There's like some sort of script, like I'm looking at some sort of ancient script with some alien alphabet, on the outside of it, right? Sort of like H shape or I shape, J shape, this thing, sigma shapes. But that's actually their pore, right? The same pores that we were looking at in the center of the valve have sort of a C shape, and down here the openings kind of have H's and S's and C's as well. But they look a little bit like little letters.
Mine of a snail thread is totally diatom scripture, and Pacific Lincoln is enjoying the secret messages in the diatom.
Yes, they are. They're like little diatom poetry written into the skeleton of the diatom, into the cell wall. So really kind of neat, and I really sort of enjoy looking at the things that we would never be able to see in a light microscope, basically, in the scanning electron microscope. So the apical pore fields that are down here just kind of have little simple holes, and it's distinguished, one, by the fact that it's at the apex, or the end of the valve. So the end, or the like the what we would call an apex.
Something that says it looks like a ripe situation for us to start a diatom conspiracy about the secret messages written on there. That sounds right up my alley.
You like conspiracies, Mallory?
A little bit, yeah. I'd say I'm probably the most conspiratorial individual here.
In my lab group?
Yeah. Honestly, that's easy money, though.
Yeah.
Yeah.
I'll just have to take your word for it.
Do you think you have more experience with conspiracies or something?
No.
Who else would it be?
I don't know. I actually was just deferring to you. I don't really know what conspiracies people might believe.
Brother Trent said, did he say Apex?
Apex, yes. Like the video game you were playing the other night.
OpenSet said ancient aliens are old news. Now it's all about ancient diatoms.
True. Why would we need to have something external? We have plenty of things that are kind of interesting that are internal, right? Inside, from inside the water of the earth. But I actually like this view a lot, and so I'm gonna slow it down and take a little picture, which is what I do when I like a view that we collect. And did you give out a shout out to OpenSet and Pacific Plankton and Mind of a Snail and District 10 mushrooms? I'm sure you did, because you're super responsible like that.
Well, hear me out. Hold on. I did just get here. How was I to know who you have and have not shouted out to?
Well, I'm just wondering. I wasn't judging.
I feel a little judged, though, to be Yeah.
I think you always feel a little judged.
We should turn this into...
Maybe you always feel a little judged.
What is micro-authority?
Micro-authority lets you pick the next thing that we look at in the SEM. Like, next time we get on and we do a little stream and you want to tell me, like, oh, you're going to look at snails next time. All right, we'll look at snails. Did somebody just exert micro-authority at us?
Greta Shrimp was asking, which is interesting because I thought she was supposed to know everything.
You did?
I did, but that's the important food.
All right, here I'm going to collect this image. It's looking good. It's got a little junk on the surface, but we'll deal with that. And then I'll pop over and see what's going on. It's still maximum.
And I get a photo of the Diatom Valves.
You don't need to exert micro-authority for that. You knew I was going to take a picture anyway.
You always... Can you read that?
Yeah.
Okay.
So Crunchy Lumpia, with extra Y's in there, said, is it true that there are dead diatoms that were once in the oceans that are now in the Middle Eastern deserts and are now oil? And this could be a dumb question. An old marine bio teacher told me this. So one of the things that's kind of cool about diatoms and other kinds of algae is that when they're living, they generate a bunch of oil, like fats and oils. And when they die, if they're buried at the right depth, what will happen is that the sort of algae-rich sediments will become something called sapropel. And if it's then buried and kept at the right temperature for long enough, it will be converted into oil. So you can find, like, where oil is found is not necessarily where it's generated. Oftentimes it will migrate out of those sediments once it becomes sort of capable of doing that. But in that case, the actual skeleton of the diatom isn't producing it. The skeleton of the diatom is made out of silica, but the living organic matter, or the once living organic matter, gets sort of converted into this sort of sapropel, which ultimately becomes oils, like the kind that we would mine as a resource. So sort of they're right. I don't know. Will I pull a tooth out so we can see it under the SEM? Dangling, that my daughter has teeth that come out, and so I don't need to pull anybody else's teeth out because hers will eventually fall out, and my wife wants to put one under the SEM, so it can happen for sure.
Yes, we are live right now.
Hi. Hello.
I'm just talking about your work, and I was going to show her the SEMs. So Dr. Stone takes most of his samples and then wait for good identification on SEM. And we do the same thing for soil macro.
All right, so have an apical pore field of Cimbella mexicana, and then it also had some of this sort of script in here looking pore, and I'm going to just zoom in and take a picture of the pore script without the rest of the apical pore field in it, just for Pacific plankton. So we can see the cool, crazy alphabet soup this diatom has made. And I'll take a quick photo of that since it doesn't really require me to do anything else. And I can come back and see what's going on in chat. Hello, headshot specialist. All teeth eventually fall out. Well, the baby teeth do. Yeah. It's Prinzikowski. Prinzikowski. Hello. So we have just the poetry in this one. Just the diatom scripture. And
No!
that was your laptop. Why did you throw your laptop on the floor?
Throw it. I'm so sorry. No! It broke!
Oh my goodness.
No!
You broke it?
No! No!
Is it broke?
Oh,
Did you destroy it?
yeah.
I don't know if it's actually broken. Your actions have unsettled everybody in the channel, Mallory. Put it under the SCM. That's a great idea, Dangling.
I'm waiting here.
Why are you blaming here for what happened?
Terrible Indiana is a disease.
Because your laptop fell on the floor? Did you destroy it?
Yeah, I'll get over it.
Prinzikowski says they can fix it.
This piece broke off. I don't want to wiggle it too much. I don't know what to do
What
about
piece?
that.
The corner of it?
Yeah.
Wow. Just tape it down. A little duct tape, it'll be fine.
Really?
How did you do this?
I moved my
You were trying to
chair.
sneak up on me.
I wasn't trying to sneak up on you.
It still runs though, right?
Yeah, it runs.
A little bit of glue, it'll be fine.
I'm just like one mistake away from having a complete breakdown. We were really close, guys. Were you
Oh.
guys scared? I was scared. Were you guys scared?
I mean,
Good time.
we're all always just one mistake from a little breakdown, I think.
If the screen is on everything fine,
I think what I would do if I were you is transfer the contents that are important to you onto something that's maybe less likely to get destroyed, just in case. And also, maybe I should put a thing in where people can use their channel points to watch you throw stuff on the ground, because they seem very excited by that part.
This is funny because it's quote day, one mistake from a little breakdown.
Yeah.
How is
Alphabet
this formed?
pores. Oh, how's it formed? Well, diatoms just lay down silica. You know, I don't really have an answer for that. But it's, you know, they're pores, so it's like part of their body, and it's part of their process of polymerizing silica and then placing it where it goes. You know, that's not a very like satisfying answer, but it is the truth. So all these diatoms that are around us that are here on the slide or stub are actually Ellerbeckia, and this is the sample that was sent to me by a total random person. Well, not totally random. I mean, I guess we follow each other on Twitter, but was just sent to me. We were hoping we would find some specific types of diatoms from it, and we've yet to find any. But we did get some nice clean views, and there's a lot of Ellerbeckia in this sample. This is the sort of poker chip margin of Ellerbeckia that we're looking at, and then internally these are girdle bands, and they form these long colonies face to face, so the valve face is here, connected like poker chips with the girdle bands kind of wrapped around them to keep them in place. And so this is one big colony of Ellerbeckia, and I'm going to zoom out so you can get a sense of roughly how much Ellerbeckia is in the sample, because there is a ton of it. And this is our Cimbella mexicana that we were looking at, And you can see that the winter time, which is when this sample was collected just like a week ago, there's a lot of Ellerbeckia living in this stream. So what's cool about that, of course, is that we get many views of the Ellerbeckia that we have in the sample, including some where we can see a lot of the internal and external structure. So we can zoom in on this little lifesaver looking guy and sort of see what goes on the inside of it and also on the outside of it.
Specifically, he wants to know if you got rid of the pink fluff.
No, I still have not figured out what the pink fluff is. So one of our sample has some sort of mysterious pink colored fluff in it. And in the microscope, like the light microscope where I was looking at it, so we got two samples. One was this one, and the other one basically had some weird sort of pink fluffy stuff in it. And we also got some rocks, so it's possible that I could also scrape some of the rocks and get some new material to look at that's not in either of these things. But the one with the pink fluffy stuff in it, I put it into nitric acid, and I thought if it was living that would get rid of it, and it should. But it's still around after that. And then yesterday when I came in for a little bit to the lab between my classes, I spiked it with a little bit of hydrochloric acid, which would normally get rid of it if it were carbonate, and that did nothing. And then I put a little bit of hydrogen peroxide in it, just thinking, well, if it's none of those things, maybe there is some sort of organic component to it. And hydrogen peroxide did nothing. So I have no idea what it is. It doesn't look detrital, like clay or silt, but it looks sort of like semi-organic anyway, but I haven't been able to figure out what it is. So my next plan is to consider putting some KOH, some potassium hydroxide on it. So I'm just working my way through things it could possibly be. One potential option is that it is a base-soluble material. Unfortunately, diatoms are also base-soluble, so if I put something in it that would dissolve silica, if I left the diatoms in there long enough, it would dissolve the diatoms too. So I have to kind of put a little bit in and then just basically stop it from digesting any bit, and if they're base soluble, it will, and they're lightly silicified, they'll get rid of it. So yes, Mallory?
Head trout specialist said, could it be a new species?
The stuff that we're looking at from Idaho is very likely a new species. The thing that we're looking at right now is Ellerbeckia, I think Inariensis. So this one is one that we know. The pink stuff is not a diatom. The pink stuff that I'm looking at, and I don't know what it is. It doesn't have any like well-defined form. It looks like blobs in the light microscope. And I could stick it in the SEM, which is an option. But I'd like to try to get rid of it first because I want to look at the diatoms in the sample, and they kind of blocks all of my view. Normally when we digest samples, especially diatom samples, our primary objective is to get rid of organic matter so that we can see stuff. So, I mean, it's not like I can't see the diatoms, but there's other things that basically cover it. And so I want to try to get that other junk out of the way, and so we can have nice clean views of the diatoms. And especially for the SEM, it becomes critical that we can do that. So, and if I can't figure out a way to get rid of it, then I don't know how I'll see the diatoms in the sample very well. So this is a nice internal view of that Cimbella mexicana that we were looking at. The central part of it is covered with a bit of junk down here. And Cimbella mexicana have a little hole right in the middle of the valve on the outside called a stigma. And on the inside of the diatom, that little stigma turns into this thing, which is like a cluster of little holes. So this is the internal expression of the stigma of a Cimbella mexicana. The little hole on the outside we'll see in another view of something, but I just sort of wanted to show you what the inside looks like. And then this is where the Raphies would end normally. There's a little piece that connects them internally, but externally they just would be little, the Raphie ends here, and the little hole is basically between them.
Pacific said enhance, please, and British Shrimp asked if it was a Mexican diatom, and then IHVH 1969 said, why?
They said what?
Guys?
Guys?
That's it. I thought there'd be more.
Just guys?
Just guys.
Oh.
Guys.
So I'm going to zoom out a little and see if I can find us an external view of Cimbella mexicana, and maybe an internal view that's not covered with some schmutz, because nobody wants junk on their pictures, you know? You get a little like chocolate sauce on your face, you don't want that to be in the photo. So all Ellerbeckia, all the time. So there's sort of got an overwhelming number of them present in the sample, and as a result it makes it hard to find anything else. But there's a bunch of little diatoms that I'm sort of skimming over by zooming out this far that we can't actually see, because I just wanted to sort of see. This is our Cimbella mexicana, the same thing we were looking at before. And if I zoom in close enough, you'll see it has the sort of alphabet pores here, right? The little C's and S's that we saw in the center. And this is the external expression of the stigma. So the thing that we were looking at, that little patch on the inside, on the outside looks like this. So it's just a little nefarious looking hole with a small depression next to it. And that actual depression is, I think, pretty common for the species, because I've seen it in a couple of views internally and externally on the slide. So this is the stigma. These are the ends, the proximal ends of the Raphie, and they usually have like a slightly expanded external opening right at the end of the Raphie, of the proximal end. So I'm gonna slap it down to a 7 and we'll take a picture. So you can still see all of the lettered looking pores on this thing. Really pretty. And several times when I've been watching Del Maximum streams, he's had Cimbella, and sometimes they're actually Cimbella mexicana. This is one of the key characteristics that you can use to tell that you're looking at Cimbella mexicana, which is that it has a little stigma that's basically placed sort of in the middle, slightly on the ventral side of the Raphie positions, and then the little alphabet characters turn into squiggly lines when they get close to it, and these sort of really prominent jaggedy wiggles that are right here are sometimes visible as kind of pronounced end pieces to the rows of the striae. So you can see them on both ends right here, all around the middle typically. So they'll have sort of like caps on the ends of the striae that are kind of these more thickened openings, and then the stigma. Boink. We'll just take a picture. Oh, hello, guys. That's what they were trying to tell you. IHVH 1969. Hello. So the question that Breaded Shrimp asked was, is it Mexican? I mean,
Rocky Feller, thanks for the follow.
Yeah, thanks for the follow, Stan Gunn and Rocky Feller. And we had a follow by District 10 Mushrooms, but we know that's our friend Ribozoid, who's just hiding under his other account that he streams from. Proximal Raphie end. Yeah. Look at you, Pandemic Watch. You're going to be a diatomist by the end of this. If you hang out in my stream long enough, you'll learn all of the ways to identify diatoms. And I mean, several of the genera, right? Sometimes even species. There's like a belly button. That's exactly right. It's a little belly button. You are lurking and you will be chilling in the Discord playing. Okay. Thanks for letting us know and for lurking. Arga Gubin wanted to know how many species of diatoms there are. We don't know the answer to that. The estimates by taxonomists are that there may be as many as 2 million species of diatoms. The ones that are described are significantly less than that. So typically, I think our normal estimates are something like there may be at least tens of thousands of species that have been identified. But I think if you incorporate all of the freshwater and marine and fossil species, we're probably looking at least like 100,000 or something described species. I don't know. But then there's a whole bunch that haven't been described, basically. So it's hard to get a handle on. There's dozens of new species of diatoms that are published, I would say, every month. And sometimes many at once. So it's hard to get a really good handle on that. For ZZ chops, are all the samples I look at dead? Well, these were collected from living material, and then I processed them to get rid of the organic material, to put them in the scanning electron microscope and to make slides out of them. But to be fair, I still have the living material actually right over there in my backpack. And so I can look at some of the living material on a light microscope, but you can't really put living material in the scanning electron microscope because it requires us to put it into a vacuum, and that usually kills things like instantly. So diatoms are no different than that. They wouldn't be able to survive a vacuum for very long. So we would usually get rid of all the like living parts so that we could actually just see the skeletons, which are what we're looking at now. So but that's a good question. So nothing in the scanning electron microscope is ever going to be living, probably. It's just not. I mean, you have to coat these samples with gold, so that also makes it very difficult for things to be living in there. Usually coating something with gold will also kill it. And the thing that we use to put the gold on it is also, it requires a vacuum. So a lot of things that are going to be like lethal for organisms. What'd you say?
We could call breaded shrimp a simp.
Oh. I don't think you're allowed to bully people like that anymore.
Yeah, so if you could, unbelievable, Nika. It's, oh, hi, Chocolate. Who's Hillary?
Hillary?
He said, morning, Hillary, morning, guys.
Who is Hillary? Did they get your name wrong again?
Maybe.
Did they were thinking that Mallory is a Hillary?
I can be Hillary if that's what you need.
It's what we all need.
Probably, can I get my VIP back after you called me Hillary?
Wait, who took your VIP away? I think you have a VIP still. It's just that, oh no, the moderator control basically kind of takes over, I think. You have to be one or the other. So.
Yeah, Mika's VIP again. Thank you for that.
This is good. All good. We'll take a nice picture of the inside of this Ellerbeckia. We're just looking at the inside of the valve. So one of the things that's nice about the scanning electron microscope, because we're not looking through diatoms, we're looking at their surfaces. We can actually tell whether we're looking at the inside or the outside of the valve. It's a lot more difficult to tell in a light microscope, but in most cases impossible to tell whether you're looking at the inside or the outside of the valve because you're mostly seeing through it. So with a little bit of careful microscopy, you might be able to tell whether you're looking at the inside or the outside. But it won't matter because you're mostly seeing through it. Okay, and it looks like it's going to be a good one. Just snap that nice picture of the inside of an Ellerbeckia. Yeah, if you become a mod, you lose your VP, but you know. Oh, they're trying to claim that, I see, that forgetting your name was just because they're old. Despite the fact that they come into the channel every time and yell your name.
It's Alzheimer's, you can't help it.
It's Dapper Dame. Thank you for the follow.
How old are you again, probably? Dapper Dane, thank you for the follow. I thought first.
I don't think you did.
I did. I'm like that one. I'm like, you're in the front of the stadium and I'm at the back of the stadium, so like you get the sound first. You know how like if there were performers all going and they were in the same stadium and there's music playing from one point in the front, the people in the front will like do the dance before the people in the back, or they'll like play the music before the people in the back. You know?
I don't know what you're talking about. Usually I'm in the front, I guess, at the stadium, so you back people?
Yeah.
Who cares what you're doing back there?
Exactly. Travel Geek does not want you to mock him, and he calls you Jeffrey, so you know he's serious.
Was I mocking you?
I was. You can yell at me for that. Sarah says I can teach her lab. Yeah, I'll do that, Sarah. I will. I'll stand there in front of them and I'll just look at them.
This is how she teaches.
They'll believe it.
She wants to be a professor one day, and this is how she teaches.
Yeah, I'll be really convincing.
Just stand up there and look at them. That's what you do.
Enzymes digesting albumin? Yeah, I could do that. I'll just,
You have to learn what all those words mean.
if it's anything like genetics lab, we'll just mix stuff and then we'll stop mixing stuff and then we'll just find the absorbance and call it a day.
Well, we keep Mallory back there because we're social distancing.
It's also just got done swirling some cow sheep stomachs. You see, that's what I'm telling you. You just literally all you do is just mix that stuff together, put it in like a absorbance machine. I forget the word. God.
An absorbance machine.
An absorbance machine.
What you do is mix that stuff up in an absorbance machine and then throw the machine on the ground.
That's all you do? How does the diatom add silica to itself?
Well, it's the same way that anything adds material to itself.
It sucks it in like a still amount.
Yeah. If I asked you how do you add bone to yourself, you'd be like, I don't know. Well, it's just sort of instinctive behavior. Organisms take up dissolved silica in the form of soluble reactive silica from the water, and then they incorporate it into their skeleton.
Maybe the real bones was the silica we found along the way. It eats too much and settles as a diatom waste.
They regularly add silica to new skeletal material. So.
Was it expected as if it was instinctive, metabolic? I think it just happens. It's not instinctive of you to take in nutrients.
Sure it is.
No, it's not.
Sure it is.
Your body to like.
You have to tell yourself to take in nutrients?
No, but I don't know if instinctive is the right word for it. Instinctive implies like a behavior. That's not really a behavior, that's just a body function.
Yeah.
Gosh darn it, how am I going to do it?
Oh, now you're going to Google it?
What dictates
Are you telling me you have to train a baby to eat?
No, I'm not saying. No.
Is that what you're trying to tell me? Because it sounds like that's what you're trying to tell me.
Some babies you have to train to eat. Don't do it right.
You mean like they spill cookies on the floor and don't pick up after themselves?
What?
What? These little processes are.
Is an inherited or innate psychophysical disposition which determines its possessor to perceive and to pay attention to objects of a certain, and then it cuts off, and I'm not going to click on the link because I don't want to talk about that.
Because why?
Because I don't want to, and I want to go back to reading the channel.
Okay.
It is behavioral if it is a body function. Back to your physiology, Mallory. Great news. I dropped my biology major before I did that, so that's not my responsibility enough.
I think it was probably a good decision based on the fact that you always want to argue with me about things of biology that you don't know.
But how? Listen, an instinct is not like just a. Your heart beating is not an instinct.
It's autonomic.
Exactly, that's what I'm saying.
But eating is not autonomic, it's instinctive.
I'm not saying that eating is, I'm saying your body absorbing and not placing and utilizing different like nutrients.
Oh, I see what you're saying.
You eat. Like that's what I'm saying. And I thought you were saying that like that is an instinct. I'm like that's not an instinct. I don't know enough definition.
But taking up nutrients is instinctive.
Eating is an instinct.
Yeah.
But your body taking the nutrients out of the food that you eat is not an instinct.
Well, I think I agree with that.
Okay, thank you. I was going crazy. Not all babies suck when they're born. Yeah.
Some of them suck when they get older.
That was a good one. Oh, I didn't see it, Sarah. Why is there a jagged ridge on the side of the diatom?
Because it's broken. Anywhere you see jagged ridges, so for example like this one or like this thing, it's because the valve has actually been broken or the colony got broken along that margin. And in this case we have a really nice view of the internal side of an Ellerbeckia, and it has these really interesting little pores right here that are stuck in the side of the sidewall that I actually wanted to see. So I just went ahead and took a picture of them, but you can kind of see them here, here, and here. But normally we're looking into the diatoms like they're a bowl, or we're looking on the outside like they're an upside down bowl. And in this one it's broken and we're actually looking like into the side of the bowl. And so it's sort of a nice, interesting view. And I just wanted to sort of see what they look like what the little pores look like that are there. These are specialized processes. These are the normal openings for the valve, like the side of the valve. And these are some little special processes that they have. My guess is that they're Rhemoportula. Like if we looked closely at them, they probably belong in that category of Rhemoportula because they don't look like strutted processes. But I could be wrong.
Arga Gubin, thank you for the follow.
Yeah, thanks for the follow.
Tropical Geek said, reflexive things are said to be instinctual because they're controlled by the cerebellum, yet there is control on the peripheral nerves, which is reflexive and not controlled by the cerebellum. The cerebellum is the old primal brain, the lizard brain, the cerebrum and cerebrain. I don't think that's how brain works. But
You don't think there's lizards in your brain?
no, I don't.
Well, I mean, it's entirely possible you don't have lizards in your brain, but
Yeah, Sarah, if you could verify that.
she is the lizard wrangler
Yeah.
that we have. Our resident lizard wrangling is all from Sarah. So she can tell us why did they call it lizard brain?
Why do they call it lizard brain?
Yeah. Because lizards don't seem particularly responsible for all the things that we attribute to our lizard brain.
I think it's just because they're like pretty big, you know?
What?
They're just like lizards, you know?
So we're gonna blame them for things?
No, I didn't say I was blaming them for being lizards. I just said they're lizards. This is what they are.
Well,
You
no one's gonna argue with you that lizards are lizards.
know? Yeah. Well, I mean,
Mhm. Oop, I found it. Here it is. This is a diatom that belongs to the genus Coccines. Coccines are monoraphid diatoms. They have a raphy on one side. This is the raphid valve. And on the other side, they wouldn't have a raphy at all. And also, typically, they have sort of like a different form. Like the valve is sort of shaped like this, with one side kind of domed and the other side pretty flat. And it's usually the raphid valve that's pretty flat, because it uses this to attach to things. And we've seen some Ellerbeckia, which are these big, long chains of things that occasionally have these things growing on them attached to the sides of them. So like this right here, right? It's attached to the side. It's like one of these little coccines. And here's another one. And you can actually see the sort of, this is just one valve, but this one has both valves. So you can see how it sort of sticks out on one side, and the other side is basically attached to, in this case, this diatom colony. And Ellerbeckia are specialists that usually live in environments that get wet and dry. And so in our case, the ones that we're looking at, these were collected from rocks in a stream. So probably rocks near the margin of the stream, where when the water flow is low, it would be exposed, and when the water is a little bit higher, they would be underwater. So they could basically persist in those conditions and do better than most other organisms. So you can see a little bit of an outline here of a cymbella is just highly dissolved. Just a piece of it kind of sitting here. What's going on back there?
They're talking about spending $2K on salmon.
Spending $2,000 on salmon?
Yeah, I don't really know where it came from. It just kind of happened.
Well, don't stop them.
And then Pacific Lincoln said snail slime, and I said I didn't use snail slime for a few days, and I got a zit. So, I mean, that's pretty.
If you don't use your snail slime, you get zits now?
Well, it's they probably completely unrelated to each other, but
I doubt it.
yeah, you're right.
Probably a witch cast a spell on your skin, and now you need the snail slime, or else you get zits.
Yeah, well, that's probably it. And then Mika said, I should know this by now, but what exactly is a valve? Why is it called a valve?
Diatoms are composed of two valves, so let me find an example for you. But the frustule is the entire skeleton, or the entire cell, and each diatom has two halves, and we just refer to half of the frustule as a valve and the other half of the frustule as a valve. So it's just a way for us to separate out the two parts and describe them separately, right? So I don't know if that answers the question, but they're distinguished by the fact that you need two of them to make a whole diatom in this case. So if you've ever looked at a clam, clams are called bivalves because they have two valves, the same sort of structure. Clam has two pieces that sort of can come apart when the organisms are dead. Diatoms are similar, except for diatom valves are not articulated. So, or they're not articulated by a hinge, which is basically how bivalves are structured. They have like a hinged component that allows the two valves to become open and closed while it's living. Diatoms have, like, they fit together like a box or a pokeball or a petri dish. The two valves kind of slide one inside the other slightly, and then they usually have other components, these girdle bands that are kind of wrapped tightly around the outside edge of both valves and allow them to kind of expand or contract how much volume they have in the inside of the valve by adding more of them. How's that?
Mika, I'm finding you a picture. Don't worry about it.
You're going to find a picture
I'm finding him a picture.
of a valve?
Yeah, he says he needs a picture. He thinks a picture. So I got you.
Okay.
I got you, buddy.
You have him.
I got you. Don't panic about it all.
Are you going to post a link?
Calm down.
Is that your plan, to post a link?
Calm down. What do you think?
Are you planning to post a link?
Yeah. I'm gonna send it.
While she's working on that, I'll show you what they look like in the actual Scanning Electron Microscope. And you can compare it to the picture if you'd like. So for this diatom, which I think is just another Cymbella mexicana, but I'm not positive of that, we can kind of see both valves at the same time, and could be used to sort of describe what we're talking about. So here is one valve of a diatom, and here is the other valve of the diatom. And if we zoom out, we'll see that we have the whole cell, or frustule, visible to us. Those lines that you're seeing here are the raphe. So here's the raphe end going around, and then it's deflecting towards the dorsal side, which is the side that's sort of like tilted away from us. So if I zoomed in on this, and you look very closely, right here, you'll see there's the stigma that we were looking at earlier, and here's the striae and areolae we were looking at on one valve on the top, or what we would call the epitheca. And it's slightly bigger than the bottom valve, which you can see all of the same features. So here's the raphe, here's a raphe, here's the raphe and the other branch of the raphe, and if we zoomed in, you'll see there's the stigma. If I zoom in on the one in the bottom, you'll actually see there's the stigma, right? So we have both valves sort of mirrored against each other. And if we look at the valve ends, you can see all of the same features we saw at the beginning of the stream. It's deflected towards the dorsal side. This one's also deflected towards the dorsal side. Here's one valve, and here's where it ends, and here's the other valve, and it's actually just covered by a girdle band right there. So, but they fit together, and if we follow this line right here, you'll see that it basically splits the two valves apart. If we were to separate them along that, we would have one valve and the other valve. So if I just keep sort of moving down the line, one of the things that you'll see when we get to this end is that the margin here's the apical pore field, here's the terminal end of the raphe. If we come down to this end, you'll see that this valve, like the height of the valve, is here where it ends.
Apple Jello, thank you for joining the Diatom.
Sapple jello? So the end of this one is here. The end of this valve is actually here. So that's how I know this is the epitheca, or the bigger valve. And this one's slightly smaller, and it basically fits inside this one, right? So the edge of the thing right here is overlapping, but this one's a little bit bigger than this one. And so I know that this one actually fits inside of that one on both ends. So it's like a little hat box or set of petri dishes, and this is the side of the diatom, this is the valve face. This is what we call the girdle view that we're looking at. And one thing that's kind of cool about this field of, or this view that we have, is that this diatom is resting on the dorsal margin, so the sort of most curved margin, and normally you wouldn't be able to see, if it were sitting on its side, if it were rectangular shaped, you wouldn't be able to actually see both valve faces. But on the side where there's the biggest curve, the dorsal margin, which is the one that's back here on the outside, for a typical Cymbella, what happens is, if you looked at it end on, they're shaped kind of like an orange slice, and we're looking at like an orange slice that's resting on the most curved part of the orange slice. So it's kind of wedge shaped coming at us, and And as a result, we can see a little bit of both of the valve faces, right? So this side is a little skinnier and that side's a little fatter, right? So it creates sort of a trapezoid. And as a result, we can see the valve face on both sides, which is kind of like a really cool feature. And it also lets us know that we're looking at a cymbella and not some other type of diatom, because cymbellas this is a primary characteristic that they have, this sort of like trapezoidal or wedge shape when you look at them in valve view, and or in girdle view. And so one of the issues is, of course, if I were trying to take a picture of this with a light microscope, is that if it's resting on one valve, the other one's going to be like on a slope, and if it's resting on this one, then that one will be on a slope. So you can kind of tell which valve you're looking at, like which one's up or down, based on which one's in focus completely and which one's only partially in focus, right? Because it would have a slope. It's hard to actually get the whole thing in focus at once. So it's a lot of words to talk about for just saying, like, okay, it's kind of like a box with two, like a hat box that you could open up, but that's basically what it is. Sometimes I think the SEM is a really great tool for sort of displaying the geometry, which might not be obvious in a light microscope, because you can see things like both valves at the same time in that field of view. So as we're clicking around, what you see is that mostly what our sample is Ellerbeckia, and then we have these Mexicana, Cymbella Mexicana valves in here, and then a bunch of really little things which are also in between. So here's one of these little things down here, right? If we look closely at it, what we see is that, let's see, we'll get it really close. It has a raphe. This the structure that you can see right there. Just like Cymbella Mexicana, it has a little widened opening in the proximal ends of the raphe are here. It does not have a stigma or any of the other features that we've seen. And if we look at the areolae, the openings, rather than looking like weird little letters on this one, they just look like dashes. Dash, 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 dash. Every one of them are dashes. And that structure is what we call lineolate. Little lines are representing the areolae, and those things are combined to make the striae. So if I were going to just guess what this is, I think the genus is very likely Navicula, which is because it's made of a bilaterally symmetrical shape. In other words, I could fold it along the axis, or I could fold this across the axis, and both sides would always match up, right? Or mostly match up. And so that tells me that it belongs in a bilaterally symmetrical group. And then it has lineolate striae, and it has raphe. I'm assuming it has a raphe on the other side, so it's probably biraphed. And that's characteristics for us.
Someone asked, so dumb question, phytoplankton, not zooplankton, right? Zooplankton food?
Actually, none of these things are phytoplankton. These are all benthic organisms. They live attached lifestyles. And so they are not phytoplankton. They are phyto. Phyto just means plants. But I mean, so they're technically kind of like plants. They have chlorophyll. They're not zooe because that's an animal. Yeah. So to answer your question more specifically, they're actually what are called periphyton. Peri means like a perimeter around the margin, and it's because they typically live in shallow water, which is usually around the margins of things. So we refer to them as periphyton. That's the sort of technical name that we would have for this group, and all of these things would be periphyton. So they live in shallow water, they live attached lifestyles, and they don't float. So for some diatoms, not all diatoms, just the ones we have in our sample, some diatoms are planktonic or can float. These ones basically live attached. So they live in flowing water conditions for the most part, and or environments that dry out. So these things, Ellerbeckia, the like very long colonies that we're seeing here with a bunch of valves basically in a row, stacked up. Like they live attached on one end, and then basically the long colonies are sort of standing off of the surface, making surfaces of rocks look kind of hairy. So if you were to go out and collect the rocks where these were actually in the streams where they were found, they would look sort of like they were covered with orange hairy like fuzz, because these things would be too small for you to actually see with your eye. You'd be able to see a bunch of them together, though, as being sort of brownish colored and hairy.
They said, I thought Periphyton was algae.
Periphyton are algae. Diatoms are algae. Yeah. So that's true. They are algae with a siliceous skeleton. A silica skeleton. So this is another naviculoid, probably navicula. It's the ends of the raphe. You can see that if we were to zoom in very closely on the striae, they're little dash shapes, right? So that is what we would call lineolate striae. And lineolate striae, two valves with raphes on them. And if we were to zoom out on this thing, again, it's bilaterally symmetrical. So you could fold it along the axis, you could fold it across the axis, and the valves would match up on both sides regardless. So if we wanted to make a snowflake that was shaped like this one, we would only need to fold the piece of paper twice. And we'd just cut out a shape and unfold it, and we would get the shape of this diatom, right? Not everything in this field of view is bilaterally symmetric. These things, for example, that are round, the Ellerbeckia, when we're looking at them in a valve view, or the valve face, they are, they're actually you could fold a piece of paper like many, many times, and then just cut like a slight arc on it, right? So they have many, many points of symmetry, or planes of symmetry that you could put through them. And so they're actually centric. Most centric are actually planktonic. So most organisms that are round like this are usually things that float in the water. And Ellerbeckia is sort of a weirdo. In terrestrial settings like these, the Ellerbeckia are things that we usually find that belong in Ellerbeckia live attached to the surface of like walls or rocks, or some of them grow in caves. Some of them grow on tree bark or on the mosses of trees. So they're usually found in places that are a little, like I said, sort of wet and dry. So not all the time wet typically. And that's a specialized type of environment for diatoms. There's only a handful of genera that can handle being completely exposed to air for a long time, and sometimes prefer those conditions. So it makes them kind of weird. So it's unusual for me to find them. Also pretty unusual for me to find a lot of them like this. Sometimes I find them in lake samples where some of the stuff from the shallow water gets carried into the lake. Or I had an idea once of potentially using them to give an indication of like rock falls, or some sort of event that would carry rocks from the margin of the lake system into the deeper part of the lake, because that's where we would typically expect to find them, either in flowing water or waves in shallow water, where where it might dry out occasionally. So the sample is kind of boring in a way. I mean, there's some really gorgeous views of diatoms that we have, but the variability in terms of like how many different kinds of diatoms we find in the sample is actually kind of small. And that's a normal process for environments that are kind of extreme. So, we have Cimbella mexicana, and then we have Ellerbeckia inariensis. And then those are the two dominant types of things we find at the larger scale. At the smaller scale, we have Cockanies, the little oval-shaped guys with raphies on one side but not the other. And I think maybe I've seen 2 or 3 species of cockanies in here. So a little bit more variable. And then there's some of these really tiny guys in here, and some of the naviculas. So these little tiny guys, like this one and these ones. They're only, maybe 10 microns or smaller. This cockanies, these are not. These are little acanthidium and samathidium type diatoms that are monoraphid and probably living attached to these Ellerbeckia in their normal life positions. Yes, Mallory?
Nothing. Definitely was just saying something interesting, and yeah.
Nothing?
Not really, no.
Okay. So I'm going to just zoom around a little bit more in this sample, and then I'm going to jump over to this next one, which is basically the same material, same sample. I just made a separate stub for it. So there was a lot of material in this, and I thought maybe I'll just take a look at both of these real quick. This one again is, even though we've moved to a new stub, it's full of the same species, right? It should be. We should expect it to be pretty similar. So here's Cimbella mexicana. Here's all these Ellerbeckia laying around. Here's another Cimbella mexicana. Another one up here. So easy to pick out once you sort of know what you're looking for. Here's one where you can look at the bottom of a Cimbella mexicana. So again, this one's laying on its back or dorsal margin. And so we can see its belly. And if I zoom in right here, we should expect to see the same structures we were seeing before. Here's the stigma. There's all the other features. We can we can see here there's a cockanies attached to the valve. Down here is the apical pore field. It's all of the same stuff, right? So it might seem overwhelming, but for me, looking at these things is kind of like picking out different kinds of, larger animals, because the SEM has sort of gotten me adapted to thinking of them, without thinking about the scale. Everything that we're looking at is basically the size of dust. And so it's kind of neat to get to the point where your brain is recalibrated to think of these things that are dust-sized particles as, much larger organisms, or the way that we would think of them. Like, I could identify them easily. Somebody asked you to, tell the difference between a cat and a dog, or a dog and a wolf, or a wolf and a bear. You'd probably be like, that's easy. My daughter could do that when she was 1 year old, right? But just because things are small doesn't mean we shouldn't use the same sort of skill sets to tell between them. And if you look at them long enough, basically, you can figure it out. It's not impossible. Just requires a little bit of patience and knowing what things to look for, right? You go, oh, that thing has this thing. I know it belongs with that group. Probably most of the people who are watching the channel could recognize any Pokemon I put up, right? So as an example, whereas I would have trouble identifying any of them. But it's the same skill set. It's like, oh, you know that one. Well, you see it enough, you know what they are. Still nothing from chat?
Well, we were bringing up some stuff about this video.
What video?
I don't know if you've ever seen it. It's called Let Me Smash.
No.
Okay.
Is it smashing things?
No.
Do any of them throw a laptop on the ground?
You would take me back to a dark place.
Oh, did I? I'm sorry.
Yeah.
I didn't mean to.
Okay. Can we put a celery stick under the SEM?
Yes?
Yeah, but it's watery, so
Yes.
someone drop a link to that video. Yeah, I'll get on that. Hold on, don't worry about it. Dr. Stone, it's just a bird, and it's been voiced over. I guess he didn't have a voice in the first place, so he couldn't be voiced over, but
A bird?
he's a bird, and he's trying to mate with another bird, but she's not having it.
Okay.
And he says, I don't know, it's just funny. I don't know.
It's a cool story.
Yeah.
Cool story, bro. Did you watch any of the interview?
Me?
Yeah.
Huh? No, I did not.
I did a nice interview with my friend Rachel, Dr. Lupian. And I need to do some more of those. I think they're fun.
How well is the evolution of diatoms preserved, and how fast do they change form and structure?
Well, that's a good question. So, I just want to get a nice picture of this stigma. I'm going to zoom in on that. Diatoms preserve pretty well. So, because their skeletons are made out of silica, it's pretty easy to get them to preserve, but over really long time scales, all you have to do is push enough water through the sediment, and you could potentially, if it's silica pore, would actually dissolve diatoms, which would be bad, obviously, for preserving them. But it makes it a challenge when you're looking at something that's like, as old as the Jurassic, and you're trying to preserve it. So on some scale, diatoms are readily preserved. On some scale, they're easily dissolved, right? So, but we have a pretty good record of diatoms, starting in the Jurassic, Cretaceous sort of period, in freshwater systems, they become a lot more common in the Eocene, and especially in the Miocene. And part of what I'm doing in some of my research is sort of exploring diatom evolution. So it's a little more challenging to find kind of complete records of diatoms from one place, so you can kind of see their actual evolution in that one place. So, like, broadly speaking, you can observe their evolution on really large timescales. But if you're talking about sort of microevolution or evolution that happens at the scale that you could readily observe it, one of the things that's neat is there are some places where that's been observed very clearly. Some of the work of my PhD advisor, Dr. Sherry Fritz, and one of her colleagues, Ed Terrio, is from Yellowstone Lake, where they actually observe the evolution of a Stephanodiscus, one of the freshwater plankton that live in the lake, evolving from what they think is Stephanodiscus niagara into a new species, which is Stephanodiscus yellowstonensis. And that transition happens in roughly sort of a 3 to 5,000 year period. But that's the completion of the evolutionary stage. So from the start, you start to see transitions happening, and with all species, there's a challenge in, like, where do you draw the line? Like, how do you circumscribe that is the species, this isn't the species? So if you were looking at, like, a character, any particular character that defines or helps to define a species, and then you see that character change, how much change has to happen before you call it a new species, right? So that's actually kind of a challenge. But they used sort of a set of characteristics to circumscribe where it has spines on the outside. So rather than spines occurring every 2 costi, every 2 thickenings, it has spines occurring every 4 thickenings. So the number of spines relative to the number of costi changes substantially. And there's, some that are in between that have, like, on average, maybe 2.5 between each spine. I mean, some have 2, some have 3, right? So it's like 2.5. And then there's some that have 3, and then there's some that have 3.5 on average, and then some that have 4. So, there's transitional stages. So you can actually see the whole evolution, and the transitional stages happen, well before that sort of total 4,000 year period. And usually what people will do is try to figure out, when does it stabilize, right? So we have a stable version where there's spines every 2 costi, and then later on we have a stable version where there's spines every 4 costi. And that whole transition takes like maybe 3,000 or 4,000 years, but there's all these transitional stages that are in between it where evolution is still occurring. So, but it's one of the few cases where we can actually look at the fossil record and then see this evolution happening over a relatively short time. And I also have 2 other records from 2 different lakes where I observe transitions happening like that, and they happen roughly at the same general time scale, like across maybe 3 to 6,000 years. It goes from what we would clearly call one species into something else that we would probably describe as a separate species. So I still haven't published those. I'm still working on publishing that information, but I've presented on it in the past, so you could find some of that information in my Diatom Web Academy talk, for example. Yeah, on that time scale, maybe like somewhere between, for a complete transition from one species to another, something on the scale of maybe 2,000 years, which is kind of instantaneous for typical geological records. So.
Wow, how do you calibrate the eras or years?
For diatoms that we're looking at on this scale, we use radiocarbon dating. So we would date samples above and below or within where we find diatom-rich units. And oftentimes we would accentuate that with lead-210 dating and potentially OSL dating, and in some cases they also use tephrochronology. So this combination of tools can be used to sort of validate the ages of those. And for some diatom records, there's also varves. So varves are sort of the equivalent of tree rings, and you can count back the years themselves. And again, you can use this as a tool to provide you with a pretty good idea of how much time has passed. So just like you could count tree rings back through time, you can count these layers in the lake record going back through time. And it provides you with one bit of evidence, and then you could do the radiocarbon dating and the tephra dating, and basically they should all converge on the same date, right? Or close to it. Allow you to create an age model. And that's how we date it. So it's actually kind of like a combination of tools. So if there's a problem with one tool, or if you're not sure how reliable one of the tools is, you would use many tools and then compare them to each other, which is what we usually do, so that we have multiple lines of evidence that would help us figure out how old something is. And then we put those together into an age model. And age modeling is usually done in modern paleolimnology. It's done by an iterative process where you put all of the information into a computer. It runs thousands of simulations, or sometimes hundreds of thousands of simulations, where if the dates are put in and the error basically would cause an age reversal where the ages are out of sequence, it throws that data away, and then it puts it back in with a different model where the error is basically on the other side. So like every time you end up with a reliable sequence where the dates are in order, like from top to bottom, then it would keep that information, and it throws out the information when they're out of order. And then it does this, like I said, thousands of times or hundreds of thousands of times, and then it figures out the most likely probability where all of the dates are always in order, and then basically it creates an age model based on that model. So it's actually really convoluted, but it's an iterative process, and in most cases the scientist, the computer makes decisions that are the most equitable, the most likely to be what is real, or closely approximates reality, right? Because it's a model, but reduces the error.
Monte Carlo tries a slightly different initial condition.
Yep, it's Monte Carlo. So it's the same type of method as Monte Carlo, where you just throw everything at it and you let the computer run simulations, and then it picks the most likely of those simulations to be, like the one that most reliably came out, or the most commonly came out kind of close to it, and it uses that to calculate error. So there's a very little sort of decision making by the actual scientist, which is generally good because it removes the bias of the scientist. So we don't really have a whole lot of like investment in what comes out. And sometimes you get dates that you weren't expecting from materials that you collect. And it's a good way for you to put the data in and let the computer decide whether, you know, which one of these dates is the right one, or which one of these dates should be used in a model or shouldn't be used in a model. So, and the errors associated with the dating can be all kinds of things. So if you're just counting varves, for example, in a lake sediment, your error could potentially get compounded the farther you get down. Like if you miss a varve, you miss 10 varves, you miss 20 varves. Well, it's not likely to happen if you've only counted 40 varves, but if you've counted 10,000 varves, it's likely to accumulate. So your error basically gets you a little farther and farther away from what's likely a reality the farther you get down into the record. That sort of error does not happen with like radiocarbon dating, where it just depends on how much mass was analyzed, and you use that to basically provide a quality control. So, and also you could potentially have errors associated with a number of other things like volume of dead carbon, and I mean it's actually a pretty complicated number of things, but most paleolimnologists who put age models together are extremely familiar with the potential for error and what might cause it, and it's considered when we put together an age model. So for really old stuff, like stuff that's older than 50,000 years, if you're looking at like you know, really ancient material like Miocene age diatoms that are 10 million years old, most of the ages are based off of a combination of biostratigraphy and tephrochronology, looking at the ages of ashes that are like volcanic ashes that are found coupled with it. So.
Me 42 says, so you study limnology too?
Yes, I'm actually a limnologist. I would say that most of my skill sets sort of overlap between ecology and limnology. I look at organisms that live in lakes, and then I look at sediments from lakes to analyze them for what the lake system was like. So I need to know how lakes function in order to figure out how do I interpret the changes I see in a diatom record. So the diatom is basically, for me, the reason I look at the skeletons of diatoms is because I understand their ecology in the lakes where they live, and then I try to apply that to the past. If you didn't study limnology, it would be like impossible to interpret a change that you see, because you wouldn't know how to, you know, how that would be reflected in a lake system. So a lot of times when I look at assemblages, I have to sort of think, well, how did that happen? Like, how did that change happen? And if you don't know anything about the lake system or how lakes work, then you're not going to be able to figure out a likely scenario for how that actual transition happens in a fossil record. It's like when people study a dinosaur or many dinosaurs, and you know, they see them, they have to know something about the kind of environment that the dinosaur was in, or you won't be able to interpret what you see in the changes in the record or groups of things you find together. So I'm technically like a geologist, a sedimentologist, a diatomist, a paleolimnologist, an ecologist, a paleoecologist, a paleoclimatologist. Those are basically my primary skill sets, but it's a lot to get at, and I'm also a little bit of a taxonomist as well. So I have to wear many hats as a scientist, and when you're working in interdisciplinary fields, that's pretty normal. A lot of us have to have a lot of skill sets, basically, that are applied to understand what we do well. Did we get a new follow?
Yes, by AJMGNTKZPN.
That's so many letters.
That's what a pronunciation was. I don't think there was a single vowel in there other than the A.
Well, hello, no vowels. Thank you for the follow.
Hmm?
I just called them no vowels.
No vowels?
Yeah.
Okay, no vowels.
Yeah.
Green Dish Chow asked a very interesting question.
What is it?
It's to me.
Oh,
Oh, yeah.
what
Oh,
is
yeah,
it?
forget about it.
What is it?
It says if you had a research project that had unlimited funding and time, where would you go and what would you research? Or maybe he just wanted my attention, so he could ask you the question.
Oh.
That would make a lot more sense. Okay, yeah. You can
My guess is that if Mallory had, I'm going to guess for you,
Okay.
if she had unlimited resources and could just do whatever she wanted, she would study dolphins.
I like dolphins. Yeah. I used to want to be a dolphin trainer, but no, the question was for you. You go ahead.
I actually right now have a project that requires near infinite resources that I want to work on.
$1 million one.
It's way more than $1 million.
A Brazilian.
It's not a bajillion.
Or Brazilian.
It's not a Brazilian. It's not a Brazilian dollars. So we have a project we're trying to do in Lake Tanganyika to core all the way through the entire record for Lake Tanganyika, which is likely to be about 10 or 12 million years, from the modern times all the way through. And the reason it's so expensive, so the estimates that we have on how much we think it's going to cost is something like $25 million. So to me, that's nearly unlimited resources. It's going to require probably several years of writing grants to acquire that kind of funding. But the reason that I'm so interested in it is because it's a single site where there's a 12 million year long record. And we could go from the modern stuff looking at samples that are basically a centimeter apart, right? And continue to do that all the way back through time, all the way back to 12 million years ago or 10 million years ago. So there's an incredible potential there to ask questions about diatom evolution and potentially answer them. And not just diatom evolution, but also fish, mollusks, ostracods, any kinds of organisms that we would find in that environment in a typical lake record. And we would be able to go back through time and look at a really fine sort of slices of time and reconstruct how those organisms evolve. And Tanganyika is actually a really important place for fish. It's got something like 500 endemic species or something of cichlid fish, and it has a whole bunch of endemic diatoms. And so for me, that would be really a fascinating study. We have some hope that we might actually get that project funded. So right now we have one grant for about $1 million that has been funded to start working on that research. But we can't really start until we have all of the funding. So, like we're 1/25 of the way there. But we have some grants that people are working on to try to acquire those funds. And if we're successful, we'll be trying to get at that information. It's also a really important record, not just because we can reconstruct, nobody cares about diatoms except for diatom people. So, the evolution of diatoms and those sorts of questions are just a piece of it. It's also important because we could potentially figure out something about human evolution through that same time interval. And that's actually really kind of an important aspect of it. We have, an area that's close to where humans, or within the area where humans evolved, and through the time range at which humans evolved. So we could ask questions about people as well, and that's something that is very interesting to me, where we can apply our reconstruction of past climate through those same intervals. So it's like a multi-pronged kind of study that would allow us to ask questions about all kinds of different organisms and how they evolve, and also how climate changed across that period. And those two things together then will be very valuable to us in science to have an answer for. So because Tanganyika is so deep, it's almost a mile deep in the middle of the lake. It's a technological challenge, and that's where a lot of the cost is. And then because we're looking at materials that require so many different scientists and so many different people would be interested in looking at the materials, we would have to collect a lot of sediment cores from that site in order to actually analyze it in a way that would be valuable for everybody. So it wouldn't just be like I get my diatom component and then and that's all that matters to me. If you're going in and you're collecting this and you're spending a lot of money to get it, you want to make sure that it's something that all scientists that would be interested in working on it would be capable of sort of getting what they need from it. So how's that for answering the question? It was a pretty cool one.
They also want to know, do diatoms contribute to nutrient spiraling?
Diatoms, when you say nutrient spiraling, what do you mean? So nutrient spiraling is like a technological term that means like nutrients in river systems where nutrients are collected and then eaten by things, and so they're like in the body of an organism for a little while and then they're remineralized and back into the environment. Diatoms absolutely contribute to that, if that's what you mean. If you just mean like nutrients spiraling out of control, like harmful algal blooms and that sort of thing, typically not. In the marine realm, you can have some diatoms that can create harmful algal blooms, but in most places, diatoms are a positive component of the environment. They're food sources, and they will collect nutrients for organisms by collecting them for themselves and replicating and then being a food source. But they don't typically create a lot of the problems like cyanobacteria or green algae do. So, and in fact, they're usually sort of the gatekeeper for those nutrients, because if you have silica in the environment, and you add a bunch of nutrients, you'll get diatoms. But if you don't have silica in the environment and you add a bunch of nutrients, you'll probably get green algae or cyanobacteria. So typically they can outcompete cyanobacteria that are harmful and green algae that might be harmful, and so as long as there's silica present in the water, those things generally are suppressed. And typically what happens is you add a bunch of nutrients, the diatoms consume all of the silica out of the water because they're consuming all of the nutrients, and then once the diatoms have basically consumed all of the silica, diatoms can't grow in the water anymore, and the other things usually then show up. So in a typical seasonal succession in a lake, you'll go like early on when the lake starts mixing and there's a bunch of nutrients available, there's a bunch of silica available that gets released in that process, and diatoms will bloom. And then as the season sort of progresses and the diatoms consume all of the silica, usually we see this sort of clockwork-like step through the rest of the algae. So as the climate gets a little bit warmer and diatoms will eat up all the silica, they'll usually be replaced by green algae, and then the green algae will consume a lot of the nitrogen, and then they'll be replaced by cyanobacteria, because a lot of the cyanobacteria can generate their own nitrogen. They can do what's called nitrogen fixation. So there's typically this sequence that goes like diatoms in the spring, followed by green algae, followed by cyanobacteria, and then, until the end of summer, and then the lake mixes again and the silica is released and we get diatoms blooming again. So like I said, they're kind of like a gatekeeper. And if you don't have silica in your environment and you add a bunch of nutrients, you end up with the stuff you don't want that creates like the scum on the surface and the nasty smell and potentially toxic conditions in lake settings. Yep.
They're just making statements, except one said, so why don't they put silica in the Klamath?
This is sort of an interesting question.
Did I say that right? Klamath?
I don't know what you're saying. The Klamath? Is that Lake Klamath? They generally speaking, how people address nutrient problems has not, at least to this point, been solved by adding silica to the system. But it should resolve the problem. And I have a former PhD student who's been working on this. He actually has access to like a sewage treatment plant where the water creates these sort of really nasty blooms. and they came and crushed up a bunch of diatomite and turned it into solution so that it would be accessible to diatoms, and then dumped that into one of these sort of tanks where they have a whole bunch of cyanobacteria blooming that are creating problems for the wastewater treatment plants. And the first year that they did it, basically, they no longer got the cyanobacteria blooms. So I think part of the issue is people just haven't figured out a way to develop the technology that would be required to do that on a large scale, like a really large lake system would require. But I think it actually is a viable solution. And I think Jace's work will eventually turn into a pretty interesting paper on the topic. He's just been busy trying to finish his PhD. So, but I think we'll see some publications eventually that kind of discuss this as a possible solution. So, coming up eventually. But it's kind of hard to like just go into an environment and dump a bunch of silica into it, because that could create its own set of problems associated with just dumping nutrients into the environment, like creating like separate environmental issues. And I think that's something that people need to be careful about. You don't want to solve one problem and create another one. So it is a question that we've, at least in my lab, we've discussed and are working on. All right, I'm going to jump over to a different sample, and this one is actually at a very different elevation, so I need to do a little bit of autofocusing to get it into the field of view and focus. And this is sort of a fun little sample. It's one that my undergraduate student, Rihanna, transferred from one of the other stubs we had onto this stub, and she used the micromanipulator to do it. So these are things she was practicing on, moving with the micromanipulator, these Ellerbeckia, from a sample. And this is what we were trying to get on the stub, and this is a Gomphanese from that same material that we were looking at, but from a different time. And this is the one that I usually call the horned beast, because I don't think it has a name. It's closely related to a known species of Gomphanese, but it's not the same species. And really I was hoping to get a valve view, like the other view, but we didn't have many specimens to try it with in the material that she had. So we're working on this. We'll probably do this where we actually reposition some of these so that we can see the valve view and get some pictures of them in a different position. But this is kind of cool because she picked this up with the tool and moved it from a different sample onto this stub as an isolated aspect of it. And you can see why I call it the horned beast. Maybe it's got these two little bumps out here that look like shark fins or whatever, but they're kind of like sticking off the top or the front of the diatom's body. So this is the head pole of this diatom, which is, as I mentioned, a Gomphanese. Pretty sure on the genus, and I know that the species kind of belongs together with a known Gomphanese, Mamilla, which also has these same horns, but has a different shape. Like its outline is not the same. So we're kind of excited about working on this diatom, and that was what the material that we were looking at before. I was kind of looking through it to see if we could find any of these things in it, but there weren't any. So I'm back here looking at this sample, and I'm just going to collect a real quick image of the horns on this one while we're here. And then I haven't taken a picture in a little bit, so I can come back and sort of see what's going on in the chat. Yeah, the Klamath River in Oregon. If you have really bad algae, yeah, that's a... potential issue. So Pandemic Watch actually brought up a really interesting idea, which is Diatom Resurrection. I don't know if you meant it to be this way, but I have colleagues who've tried to do this where so diatoms can insist and then can be brought back to life from very long dead stages, or what we think of as being dead, but they're just basically like hibernating, a little bit like water bears do, or other things where they basically go into a sort of hibernation state. And so I have colleagues, David Burge and Mark Etland, who've been working on trying to take material from a core from hundreds of years ago and basically pull out a bit of the material and add it into a nutrient-rich environment and bring some of those zombie diatoms back to life and resurrect them. So there's a whole field in limnology known as resurrection ecology, and we're sort of exploring this as a way of trying to understand a little bit better what their ecology is. So we could find diatoms and bring them back to life from the sediment, and then grow them in sets of different kinds of cultures to try to figure out what their preferred environments are like. Yes, you can actually do 3D scanning on these. There's a couple of different ways that you might do that. One would be to use an atomic force microscope. I don't have one of those. They're very expensive relative to what I have. Fresh Cow, we're looking at a diatom from Idaho, from a stream in Idaho, up close in the Scanning Electron Microscope. This is the skeleton of it. It belongs in the genus Gomphanes, but this one has horns. So it doesn't have a name. I think I've been working on trying to describe it. So it's Gomphanes SP right now. Yeah, I just call it the horned beast. For Zitmus, you could image diatoms from many different directions and then use that to create a photogrammetry model using photogrammetry. It's the same way that they do with on Google Earth, right? The satellite flies over and it takes many pictures, and then you have sort of these different angles of the same landscape, and then Google Earth will let you sort of see things in 3 dimensions. So you can use the same process in a scanning electron microscope where you image things from many different directions, and from there you could create a 3D model. And I've actually done that. Hang on a second, I need to type this up. And I'll show you. So it's actually underneath my camera. Apologize for fumbling the camera a little bit. But this is a 3D model that we made of a diatom by taking pictures from many different angles, and then ultimately turning that into a 3D model that you could print on a 3D printer. So in my case, I think I used about 80 images from the scanning electron microscope. So we turned the stage every 20 degrees. I would take a picture, and then I would tilt it a little bit more, and then I would offset it by 10 degrees, and then I would take every 20 degrees, take a bunch of pictures, and then I would tilt it a little bit more and do the same thing. And because the controls on my scanning electron microscope are, Pandemic Watch says take care.
I was going to say goodbye to you on a roll, but I didn't want to ruin your talk.
Take care of your laptop too.
I know.
Yeah, so pretty awesome in order to capture all of the detail that we wanted. And I actually have a little bye. So I actually have 2 of them that we printed, and then you could glue them together to make a whole diatom if you wanted to. So that's what this is sort of sitting on. And now that Mallory's gone, I can take my mask off. So we can do that and actually create 3D models. If you wanted to print one of these, if you had a 3D printer and you want to print some 3D models of diatoms, I've actually posted these files on my Twitter account before, and I can probably dig them up and put them, maybe in our Discord or someplace, if people wanted to print them. They're kind of fun. And this is a species of diatom that I described. So this is kind of cool. I've got a 3D model of a diatom. This is Stephanodiscus coruscus, a diatom that I described last year. So we made the model 3 years ago, but it took me 3 years to get my act together and describe it as a new species. So now we actually have it in, if you were interested in holding a diatom. Normally they're dust-sized. So this is mostly what I use for outreach purposes, so people can kind of get a sense of what they look like. So send some to Dr. Abel at HSU. I don't know who that is, but maybe send me their link. Yeah, so it's a pretty cool idea. And then I've also had students make models by just building them in modeling program tools. But I actually kind of think this is cooler to be able to use real diatom images to make it so it's much more realistic and a little bit lumpier, but also kind of cool, I think. And for this, we actually have a skinned version of it. I can showcase that one of these times. I probably should have it, but I have a little video of me spinning it basically in the 3D drawing program with the skin that was created by a composite of a couple of pictures, so that you could see it from all the different angles. In it looks like a SEM model, right? That you could just spin and see the whole diatom. So it was really cool. It's sort of a collaboration between me and my colleague Alex Badillo, who's here in our department as another scientist in our Earth and Environmental Systems. They actually study anthropology, so they're not diatomist at all. And he was kind of freaked out by me giving him a bunch of images of these really tiny things, because he normally images landscapes and does this sort of thing, or pottery, right? Something that you might want to preserve for a museum. But it was kind of fun to be able to do it and then and skin it, and so we have it as a model. I meant to do it a couple of other times. It's a kind of a lot of work to image things from, take that many images of them, and then to get it to work inside Agisoft, which is the rendering software that does the photogrammetry. So, and I don't have most of those skill sets, so I just take the pictures and then hand them to somebody else who does all the sort of challenging parts with the modeling. And also, they're a lot better using the 3D printer than I am, so they actually do all the printing as well. So this isn't a version of this Gomphanes that we found at the site, and here's a bunch of these Ellerbeckia that we were looking at before, and my undergraduate student, Rihanna, transferred these here, and then she actually smashed some of them. And I think we actually have, I think this is actually, this is Costinodiscus that we transferred onto the stub from some material that Pacific Plankton gave us. So she picked up one of these Costinodiscus and brought it over. And then when she went to set it down, she smashed it. So a little bit heavy-handed on the placement when she was putting it down on the stub. And as a result, it's a little smashed up. But diatom's still pretty clear to see. It was from some of the live material from December, I think, that Pacific sent me. And we were just trying to move stuff, so we wanted to find big diatoms we could move. So here's a couple of them. I think this is a transferred piece of a Thalassiosira here. You can see these really interesting little sets of pores that are lumped together on it. And then I think there's another one over here that we transferred. Yeah, this is another broken one that got picked up when we moved the other specimen. And I think this is a cosinodiscus. I'm not positive about that, but the pores look right. So we were, oh no, that's a Thalassiosira. Those are right there. Those are strutted processes, so Thalassiosira. But so we have a couple of these on here. I think that will also be one that she transferred that's marine. Yep. So some more marine species that we transferred onto this this stub. And the funny part is, so we have these things from Idaho, and then these things over here from the marine stuff. In fact, actually, I think maybe this might be Ellerbeckia from, I don't know which one this Ellerbeckia is from. This might actually be from the Idaho stuff, or it might be from Pacific Plankton's material, because she also has some Ellerbeckia. This is probably that's the freshwater version. But, you know, here we have them smashed together on the same stub like you would normally have. And then I was trying to flip this diatom, another version that we have it over so that we could see the other side of it. And in the process of doing it, I noticed it started to crack. So I just went ahead and smashed it a little bit while we were moving it. So I was hoping I would be able to see the inside view of the diatom, but it turns out they keep breaking along the raphe rather than breaking like open into two halves. And so we haven't actually been able to get a lot of good views. So we ended up with like a broken piece, and then this junk associated with it that kind of falls all over the stage. Here's one. Here's a bit of the inside of one of them. So you can kind of see what they look like on the inside. But the image quality, well, the image is fine, but the you know, we don't have the whole thing. We just see like a little snippet of it. So I don't have a really good sense of what it actually looks like. But I think if I get this in focus and maybe we zoom into one of these little holes a little bit, we might be able to see the areolae from the outside of the diatom through one of these little windows. Oops, I took it too far out of focus. So I don't know. I think if I do this and I hit auto brightness contrast, it'll give me the brightness inside of there. And now the whole thing is super bright, but I can kind of see into these little windows of darkness. And I think if I slow that down a little and focus in there, I might be able to get the striae to show up. Maybe not. They're big holes, so we're super bright. Oh, you can kind of see them in there now, right? They're really big holes, so it's kind of a challenge to get them in focus at a different height like that. Maybe this bigger one, if we zoomed in on it. Not working out super well, but I could slow it down and we could kind of see into it. There's some really fine openings on the surface of this that I can kind of see. So I'm just trying to like get them in good focus for our field of view. It's not working out super well. So in any case, this is sort of where we are trying to get a reasonable picture of the inside of this beastie, and not really managing to do a great job of it so far. So I still probably need to do a little bit more work trying to get that sorted. Maybe have Rihanna give it a shot with some of the more, a little bit more of the material that we have, and see if she can manage to get some more of them transferred into the stub that we could then try breaking or maybe turning a little bit more gently and see how we do with imaging the inside of them a little bit better. So let's go ahead and take a picture of this, I guess, because it won't hurt to have it, but it's not exactly what I want. Okay. Hey, Mama Bonbon, how you doing? Let's see, because Mallory left, I don't know what's going on. Let's see. Diatoms, yeah, I smashed it. Diatoms have a color when they're living because their chloroplasts have a pigment. And that gives them a sort of golden brown color, as specific plankton says, sort of down there below. But the skeletons of the diatoms are made out of glass, like opal, right? Not technically glass, but like opal and silica. And so they would be colorless. And so everything we see in the scanning electron microscope right now, if it's a diatom, normally would not have any color. And in fact, it would have a little bit of color because I've coated them with gold. So they would have like a gold sheen to them. Just a few nanometers thick layer of gold on the surface of them. But they essentially would be glass. So... Oh, you mean like drawing it very slowly makes the text seem ancient? Fresh cow? Yeah, that's because it scans across the surface. And when the electrons bounce off of it, it sort of captures how many. And each one causes a photoreceptor to brighten a little bit. So at the very, very fast way that it's doing it, it's actually moving across the specimen just a little tiny bit at a time, pixel by pixel, putting in the brightness for that pixel. And that's actually what you're seeing. It's not taking a picture like loading it, it's actually building the picture. So you know, it of course seems like, oh, it's taking forever to take this picture. It's an ancient technology, like a web page loading from the 80s, but it's actually the opposite. There's no picture before what you see. It's building it while you're watching. So it's drawing it basically as you're watching, and it's moving at the speed that the beam is moving. And the beam moves slowly because we're getting a high resolution. So the slower the beam moves, the more clear the picture is. And you can see that when I change the speed settings. It looks kind of grainy, and then I can move around real quick. That's because it's just scanning across the same area at a much faster rate. So it's kind of like a interesting, you know, it looks like it's old. It looks like it's ancient technology, and especially because it produces black and white images. And you think, oh, you know, like it's old and it's taking forever to draw an image, but in fact, it's black and white because there's no color. It's using electrons, so nothing you do is going to change that. There's no light. Without light, you don't have color. So, yeah, it's like a consequence of the tool rather than. I mean, you couldn't make color images from black and white, you know, from photoreceptors, giving you intensity, essentially, because that's what you're recording. So it's not like you could do better. You could do better, like, you could get a higher resolution image out of it, and you could potentially do better by getting it to scan more quickly. And the really high-end, brand new SEMs, have like ultra fast scanning capacity, and so they move a little faster and have really high resolution, but you know, fundamentally they're not, you know, aside from just being a little bit faster, they're fundamentally not much better. Like the image is still going to come out black and white. It might be a little bit higher quality, but it won't be color, and it'll still take a while to build. It just won't take quite as long. So just to give you like a sense of it, but I don't have like a spare $1 million to throw down on an ultra fast scanning electron microscope. This one's the one I have, and I rather like it, even though it's a little slower and a little bit lower resolution. But I think we still managed to get some really great photos from it, and it's highly functional. So, and also cost a ton less money. So, I will take those sacrifices if I get a good diatom picture out of it. I mean, that's all we really need to be able to manage it for my work. We got a new follow. That was Underground Rebel 10. Thank you for the follow. For people who are just joining or who've been here for a while, it often takes me a while to come back to the stream and see what the chat's saying, because I have to sort of manage the SEM. And so oftentimes I have somebody here hanging out that answers those questions or relays them to me so I can kind of talk and work at the same time. But not right now. So, hello 3D Prima. Thank you for joining us today. And also Two Chooks, it's nice to see you. Welcome in. Yeah, there's a scale at the bottom. I'm trying to figure out what's going on back here. Kind of missed some important aspect. AI coloring. I guess you could do that, but it's I'd rather do it myself and pick my colors. So I'd rather have the control as an artist to select what I want, rather than just have the AI do it for me. Maybe I'd have a different opinion if I had an AI that was doing it, but I sort of feel like I do a pretty good job of picking out the colors, in my opinion, and I like to play around with it a little bit. So here you can see on this Gomphanese that, wait, it's a Gomphanema? Well, whatever it is, it's got stigma. Here's the raphe ends in the middle here. And then we're looking at a cross section of it. So here's where I broke it. Here's where I smashed this one. And you can actually see the internal structure of the valve, which is actually a good reason to smash diatoms, because now I can see what the shape of the cell wall looks like. Here you can see it's got a really thickened component with these sort of like costy pieces that are coming up, and then they're overlain by a sort of little a thinner sheet of silica that has little holes in it. So in a sense, smashing it actually helps a little bit, because if I only saw full valves, I might have a hard time trying to figure out, well, one, what's the inside look like, and two, what's the structure of the actual wall, what we call the ultrastructure. So you can actually see the ultrastructure pretty clearly in my smashed diatom. And I had some, oh, thank you. I got a new follow there, I guess. That guy DTD. I actually had some colleagues when I was younger, and I was just learning diatoms, that would tell me, oh, once you get stuff and you've already analyzed and you've seen the external views, just like take a pencil eraser and just sort of smash the diatoms a little bit, so you can kind of see what the ultrastructure of the cell wall is. Because it's important for being able to characterize them, to be able to see those kinds of things. We need to be able to see what the insides look like. I wish I would have gotten a little bit better image of this. Instead, I've got fragmented right where I need to see it, right in here, which is, I want to see what the internal view of the, that's the Gomphanese. I want to see what the internal view of this process is, and so I need to kind of flip that piece over. So, I mean, it would be useful if I maybe put this back on the stereoscope and tried to flip this little piece over so I could see the inside of the valve, because it looks like the whole valve face would be preserved here, and that's what I need to see. I need to see the surface. I can't see the inside. So maybe I potentially could clean off some of this junk and also see this valve face because it looks like where I broke it, I might actually be able to see if I could get this stuff moved. So I'll play around with it, or maybe we'll just try to smash another one. I thought maybe if I smashed it just the right way, I'd be able to see stuff, but it doesn't look like I'm able to see anything that I necessarily wanted to see. So let's see, we've been sort of working our way around the clock here, coming backwards through samples, and I need to adjust the height again because it's focused on the wrong place. And I forget what these are, what are in these samples. I think maybe they're ones that, oh yeah, this is the ones that I treated with nitric acid, and then basically they didn't get completely all the nitric acid out, so a little bit of the nitric acid residue is like coating over everything. So this is one that, you know, if I had a couple of diatoms on here that I wanted to trash, move over to their other stub and then smash them, this would be a good one, because a bunch of the stuff that's on here is not going to be very well preserved, because we just have a little parts where the acid residue isn't present on the stub. So this might be a good target for me to smash in the future, or to have Rihanna smash some in the future. There are some diatoms on here, these little guys right here, which are kind of cool. These belong to a genus called Grunauia, and I don't know if it was recently resurrected or if it always was a new genus that was put together in the last couple decades. But you can find that one on Diatoms of North America, and I think the species is actually on Diatoms of North America. In that group, it's sort of the only straight, skinny version of Grunauia that's on that webpage. And then they're all over the place in here. So all of these are Grunauias, and there's some Ricosphenia in here as well. So the Grunauia are actually characterized by the fibula. Here's a little broken version of one of those where you can see the fibula extending onto the valve face. So this sort of structure that's here is the fibula. I'm going to try to get it in a slightly better focus for us, and also my brightness is way off. But these sort of structures that are right here, these things, don't occur in Knitsia, but they do occur in Grunauia. And if they went all the way across, but these ones only go part of the way across, then it would be Denticula. So it's like a genus that's in between Knitsia and Denticula, and got its own genus name as a result, which is Grunauia. And we have a bunch of them in here. And among the Knitsioid diatoms that exist, Grunauia is actually my favorite one, favorite genus, because they're really kind of unique looking, and especially in the light microscope. So there's a Ricosphenia right there. So I could recognize it very quickly because they have a curved profile. So this is a girdle view, and you can see that it's like curved like a banana, basically, a little tiny one, but it's laying on its side. So if we were looking at it on the valve face would be the part that's curved. These also occur in flowing water, and are pretty diagnostic of either water flowing, or like wave action usually. So if you get a lot of them in your sample, you know that your sample is probably a flowing water material. So I've got several of these samples that are covered with a bit of junk and have the same thing. So there's Ricosphenia, there's Grunauia, and Cockanice, which we saw on the previous sample as well. So here you can see the consequence of not rinsing enough of the acid out of the material, which is basically crystallization of a little bit of that acid. And if we chase it around the slide or the stub, I bet I could find some of the things I'm actually looking for kind of buried under this pile of junk. So it's just like a small error in processing. I just didn't realize that there was a little bit of residue left in there. I'd rinsed it a few times, and usually that's enough to get it out, but it's a really concentrated acid that we used, and so just having a little bit of acid is still enough to cause it to leave a residue like this on the sample. So here's some actually right. Let's go back. This thing, that's that diatom we were looking at that I smashed right there. You can see it nested inside of a bunch of pile of stuff, right? So that's actually, I think, the Gomphanese, not the Gomphanema. But that's a characteristic outline for the genus, so I can tell. And this is an Ellerbeckia that's been buried underneath this pile of junk. So I don't even need to see it, I can just sort of see the outline, and I can tell. For most things, if I've seen enough of them, I can kind of tell what they would be relatively easily. Here's one of them that didn't get buried. So again, similar sort of shape. This one's on its valve. We're looking at the valve face. And I took a picture of this one in the last stream, but I didn't get a close-up of the head. So I'm going to just zoom in on the head pole a little bit. It's slightly deteriorated, but we could at least see what's going on up here. And this one's a Gomphanema, not a Gomphanese, or probably a Gomphanema. So I'm just going to snap that one real quick while we're here. And let's see, what's going on down here? Got a whole bunch of smiley diatom mustache faces, happy little diatoms. Yeah. Oh, Cinnamon, hello. How you been? Go scroll up and see what I missed. It sounds like Pacific is handling my chat questions as usual, so that's good. Supervised learning, yeah. And also, Braided Shrimp is back from playing games. And it looks like Pacific Plankton is nailing all of the links for diatoms of North America that you might want. If you want to learn a little bit more about diatoms or about those genera that we've been talking about. So, thank you for the follow, Arc. It's nice to see you in here again. And just going to let that roll over it, take a real quick look at it, and then I won't get any better than that. The frustral itself is kind of broken a little bit, but I'm just going to go ahead and take that picture because it will be at least some idea of what the head pole looks like. And let's see, what's going on? It's getting close to 3:30 or 3:15, and so I should start thinking about rating somebody and ending the stream. Got a whole bunch of potential targets today, it looks like, for people that we could raid. So I need to pick someone, and could be any number of people. Pacific Plankton getting ready to go. All right, good. I'm getting ready to go as well. So we'll call it a day. Maybe I'll go eat lunch too, because I haven't eaten anything yet. I should say thank you to all the people that followed us today and hung out with us. So Arc, that guy, Underground, the amazing Wolf Skeptic, AJMG, R Robila 51 just followed. Thank you for the follow. Sylvia Peterson, Marvel Me, Sapple Jello, Arga Gubin, Dapper Dame, Rocky Feller, Stan Gunn, Direct 10 Mushrooms, or District 10 Mushrooms, who is just Ribozoid and his other guys. Those are our follows for today. So thank you, people, for hanging out. And we'll probably be back on Saturday, maybe with some more diatoms or something else. In the meantime, you should check out other microscope streamers. If I don't know if this has been posted. But Pacific Plankton will be on, let's see, today's Wednesday. She'll be on tomorrow night, probably, around midnight. And also Del Maximum should be on tonight, sometime around midnight. OpenSet, who was here at the beginning of our stream, who knows when OpenSet's going to be on. He usually just appears like a wild animal whenever he feels like it. I think I saw Spider ID on earlier yesterday in the morning. And then Freckled Science also may be streaming later tonight. She's on, I think, almost nightly now. And District 10 Mushrooms, who might be on around all these other fellow potential microscope streamers who you could catch. So I want to say thanks to Pacific Plankton for hanging out and for handling chat, and especially once Mallory was gone, but also helping answer questions. So let's see, we should raid somebody. How about we pick somebody different than what we usually do? We will raid Professor Birch, who is not a science streamer, they're an art streamer. But they do drawing in usually Pokemon characters or something. So I like to mix it up sometimes and bring some art people in, because I also do a little bit of art myself occasionally. So maybe we'll raid Professor Birch, and you can hang out with him and ask questions and check out some of his artwork. It's a pretty cool stuff that he does. So, yeah, we'll catch you all again. I'll probably do a little night stream sometime this week as well. Maybe tonight or Friday night. We'll see. I've got some raw materials from these samples that we've been looking at, and I'd like to throw those up on the microscope and see if we can find anything different. So, playing around with that. All right, so thanks for everybody for hanging out, and had a nice kind of quiet, casual afternoon session, the way that I like to have them. And I'm gonna go. I gotta grade some exams, so that's the rest of my day. So, all right, bye everybody, and let's go get Professor Birch. Let's attack him. All right, let's go.